Good afternoon, learners. I welcome you all again to my session. Today's topic of discussion, molecular techniques used in plant biotechnology. Now, in some of my earlier sessions, I discussed about the genetically engineered plants, their utilities, and the biosafety issues connected with the genetically modified plants. Now today we will be discussing on what are the molecular techniques that you used in plant biotechnology. What are the molecular techniques that you use during the preparation of the genetically modified plants or after the generation of the genetically modified plants. So we will take a look at some of these techniques one by one. Now the techniques that we are going to discuss today: southern blotting, northern blotting, western blotting, DNA fingerprinting technique, DNA markers, RFLP and RAPD, restriction fragment length polymorphism, and randomly amplified polymorphic DNA, polymerase chain reaction, reverse transcriptase polymerase chain reaction, which we also call as RT-PCR, DNA sequencing, and ELISA. ELISA stands for enzyme linked immunosorbent assay or analysis, whatever. Now, so these are the techniques, and these are all included in BBYET 143, that is Economic Botany and Plant Biotechnology, which is included in Unit 13 of Block 4. So you will find all these techniques in this particular unit, and I'm going to explain these techniques one by one. Now, before going into the actual subject or before going into the techniques, first of all, I would like to mention that once you develop a genetically engineered plant, then you need to analyze whether you have constructed the genetically engineered plant in a proper fashion. That means you need to undergo certain molecular analysis. Now, what do you mean by molecular analysis? If you remember that when we construct a genetically engineered plant, the first thing is we need to clone a gene. And therefore, the gene of interest has to integrate within the genome of the transgenic plant. Number one. Number two is, after the gene is being integrated in the genome, the gene has to express itself. That means the transcript has to be generated. Third, even after the formation of the transcript, the protein has to accumulate or the protein has to be synthesized. Okay, so there are three steps. First, you need to check whether the gene has been integrated. Second, whether the gene is being expressed. And third, whether the gene is actually able to translate. We call this process as translation. That is where the protein is being synthesized. So ultimately, whether the protein has been accumulated or synthesized or not. So for these three steps, we have three techniques which are listed in the very first 3 points, that is southern blotting, northern blotting, and western blotting. So now we will take the technique one by one. Now first is southern blotting. Southern blotting, this is a technique which was given by Edward M. Southern in 1975. So he was the discoverer of this particular technique, and after his name, this technique is also known as southern blot or southern blotting. Now what do you essentially do in this southern blotting? So once you have generated a transgenic plant, you need to isolate the genomic DNA from the transgenic plant. Before that, let's see what are the utilities of this southern blotting. Number one, it helps us to identify the copy number of any endogenous gene in the genome. Number two, detection of integration of a foreign gene in the genome, mapping restriction sites around a single copy gene sequence in a complex genome, evolutionary relationship among organisms, determine the genetic alterations causing a mutation. So these are some of the utilities or some of the applications of the southern blotting technique. So here you can see the steps. Now what you do, in this case? How do you undergo a southern blotting experiment? Like, you need to know how many copy number the gene is present, whether the gene is present in a single copy or whether the gene is present in double copy or multiple copies. And as I already told you, whether the gene which you have cloned is ultimately being integrated within the genome of the recipient plant, that is the transgenic plant. Because if the gene is not there, then you will not get the expression. If the alien gene or foreign gene is not there. So you have to first confirm whether the gene has really entered into the recipient plant. So for that, what you need to do, first of all, you need to isolate the genomic DNA from the transgenic plant or from any plant. Then you need to digest the genomic DNA with restriction endonuclease enzymes. So there are many enzyme sites. There are some common restriction enzyme sites within the genome. For example, EcoR1. This is a very common restriction enzyme site. So you can use this enzyme, or you can use even two enzymes, two restriction enzymes simultaneously to digest the genomic DNA. So once the genomic DNA is digested, next what you need to do is you need to check whether the digestion is complete. So you need to run an agarose gel, or you need to undergo agarose gel electrophoresis. So you need to load the genomic DNA or the restriction enzyme digested genomic DNA in the agarose gel and then run the gel. So once the gel is run and you stain the gel with ethidium bromide, you will be able to see fragments because the genomic DNA is digested into fragments. So several fragments. So now what you do is you need to transfer this DNA, or you need to transfer the digested genomic DNA onto a membrane. That is, it can be nitrocellulose membrane, it can be nylon membrane. So you need to transfer the DNA onto membrane, and there is a particular technique, or there is a particular way by which you can do that. I am shortly coming to that. That is capillary transfer. So now after the DNA is being transferred to the membrane, we call it a blot. That means the membrane is the support where the DNA is being transferred. So now what you have the membrane with the DNA. And then what you need to do is you need to add a probe. What is meant by probe? For instance, you want to check the gene of interest. So what you do is you need to radioactively label a particular gene sequence. So normally we use alpha 32P as a radioactive isotope, and we label the DNA sequence or the gene sequence. So you prepare a probe, and with the membrane what you do is you first add some hybridization pre-hybridization solution, okay, and you need to add a competitor. Why you need to add a competitor? Because you want to restrict the non-specific binding. So the competitor normally which you use in this case is the sheared salmon sperm DNA. So you use it to block the non-specific binding, and then after that what you do is in the hybridization solution you add the probe. So if there is a complementarity, or if the gene is already there in the genome, then this gene sequence or the probe, it will actually bind to the genomic DNA fragments. It will find its complementarity and it will bind, and therefore upon the development of autoradiography, that is when you expose it to X-ray plate and then you go for the autoradiography, you will find that there is a binding. That is, the probe has actually bound to the particular sequence in the genome, and therefore you will be able to confirm that the gene of interest has been integrated. So this is actually the principle of the southern blotting. So first of all, now there are certain points which need to be remembered in this connection. There are certain techniques which need to be followed. Certain protocols need to be followed. Like number one, large DNA fragments greater than 10 kilobase pair, they require a longer transfer time than short fragments. To allow uniform transfer, the electrophorese DNA is exposed to a short depurination treatment. That means after the gel run is complete, now you need to process the gel. Before you transfer it to the membrane, you need to process the gel. For instance, initially you need to treat the gel with hydrochloric acid for depurination. And after depurination, you expose it to alkaline hydrolysis at the depurinated sites so that it denatures the fragments prior to the transfer. So this denaturation step is very important in presence of alkali. Sometimes we need to neutralize it. Sometimes we need to treat it with a neutralization solution after hydrolysis. An alternative method uses positively charged nylon membrane which remove the need for extended gel pretreatment. So you can go for nitrocellulose membrane or you can go for the nylon membrane. In this case, the DNA is transferred in native form and then alkali denatured in situ on the membrane. So this completes the transfer process, or rather I would say the first part of the transfer process, where the processing of the gel is complete. Now, during the transfer process, while you are now transferring the DNA from the gel to the membrane, what are the steps that you need to follow? Now, you have the agarose gel now, where the DNA is, you have run the DNA. Okay. So the agarose gel is now mounted on a filter paper wick which dips into a reservoir containing transfer buffer. So the transfer buffer which we normally use in this connection is SSPE. We call it a saline sodium phosphate EDTA. So we normally use this, or sometimes we use SSC, that is saline sodium citrate. So this is actually the transfer buffer. The hybridization membrane is sandwiched between the gel and a stack of paper towels which serves to draw the transfer buffer through the gel by the capillary action. The DNA molecules are carried out of the gel by the buffer flow and immobilized on the membrane. Initially, as I told you, people used to use mostly nitrocellulose, but the nitrocellulose membrane has some drawbacks. That is, it is fragile in nature. And therefore, nowadays, mostly we use the nylon membranes, supported nylon membranes, which have greater binding capacity for nucleic acids in addition to high tensile strength. So this picture will show you how you need to arrange all the, all the materials when you are undergoing such transfer process. So you see at the extreme lower part, you have a tank or you have a reservoir where you have the buffer, that is the transfer buffer which I am talking about. And then there is a support or there is a platform, and there is a paper wick which is above the platform, and then on top of that you have the gel. Now above the gel you have the membrane. Membrane means where you need to transfer, that is from the gel to the membrane you need to transfer the DNA, and therefore you need to prepare a blot. Above the membrane you have 3 sheets of filter paper. Above that you have stack of paper towels or tissue paper, you can say, or paper towels, we call it in a better way. And then you put a glass plate, and above the glass plate you put some weight. That is in the common balance. For the common balance you have certain weight. You can put those weight. And then you just put this entire arrangement or put this entire setup, it may be overnight. So the next day when you come, you will see all these materials are wet, because from the reservoir there is a suction and there is a capillary transfer, okay, because you have given the paper wicks which actually will absorb the buffer, and in the process, this capillary transfer process, what happens is from the gel to the membrane there is a transfer of DNA. So the next day when you come, maybe overnight you leave it as such, or maybe for 18 hours or even 24 hours you may leave it as such, and the next day, okay, you have to dismantle the entire apparatus or dismantle the entire system, and then finally you will be able to make out that from the gel the DNA has been transferred to the membrane. So this actually completes your transfer process. So I hope you get the point that how the DNA has to be run, okay, into the in an agarose gel, and how from the agarose gel to the membrane the DNA needs to be transferred. So now you can discard the gel because you have the DNA in the membrane, that is nitrocellulose membrane or nylon membrane, whatever you are using, and you have the blot now. Now what you need to do, you need to do the next step, that is now you want to check whether your genomic DNA or whether your fragmented DNA has the gene of interest. So now what you have to do, you have to prepare a probe. Preparation of probe is itself another task where you need to actually incorporate the radioisotopes into the DNA. That is, you need to label the DNA. We call it radio labeling. You need to radio label the DNA or the gene of interest by using mostly alpha 32P. And we call it, mostly in this case, we call it a random labeling. And then that becomes a probe. And then what you do, you take the membrane and you put it, or you apply pre-hybridization solution. And in the pre-hybridization solution, you also add the sheared salmon sperm DNA, which acts as a competitor DNA, because it will block all the non-specific binding. And after that, you wash it, and then you again apply the hybridization buffer, and now you add the probe. So if there is significant complementarity, the probe will be able to bind to the gene of interest which is present in the fragmented genome. And thereafter, you need to develop it. You need to develop the X-ray film, expose it to X-ray film, and develop and go for an autoradiography. So the next steps after transfer, the nucleic acid needs to be fixed to the membrane. Before going for the hybridization, another step has to be done. That is, you need to fix it to the membrane. And here we use two techniques. One is oven baking at 80 degrees centigrade is the recommended method for nitrocellulose membrane. But due to the flammable nature of nitrocellulose, it is important that it is baked in a vacuum oven. Alternative fixation, which we more commonly use in the laboratories, it utilizes ultraviolet cross-linking based on the formation of cross-links between a small fraction of the thymine residues in the DNA and the positively charged amino groups on the surface of nylon membranes. So we normally go for the UV cross-linking method, which means that the DNA which you have transferred to the blot or the membrane is permanently fixed to the membrane. It will not wash off. It is permanently fixed to the membrane. A calibration experiment must be performed to determine the optimal fixation period. That means you need to standardize. Mostly the cross-linking, it works maybe if you go for 2 minutes, it works. There is substantial cross-linking. There is no problem. Now what you do, as I already told, I am just repeating once more, membrane is placed in a solution of labeled RNA single-stranded DNA, which is complementary in sequence to the blot transferred DNA band or bands to be detected. Since this labeled nucleic acid is used to detect and locate the complementary sequence, we call it a probe. I already told you that the radio labeled sequence, you call it as a probe. Now, in this connection, I would mention another important point, that you need to choose the hybridization condition. That means under what condition you are carrying out the hybridization process. This is very important, because you can carry it out under very low stringency condition. For instance, if the copy number of the gene is very low, you need to carry it out at low stringency condition. That is, you don't apply too much of high temperature. But if it is a high copy, then you can go for high stringency condition. That is, you can apply temperature during the hybridization process. And after hybridization, it needs to be repeatedly washed, because you need to wash off all the non-specific binding from the blot. Otherwise, it will give a dark background. You will not be able to see the clear band. So you have to repeatedly wash it off to remove the non-specific binding. Okay, and in those cases, you can undergo a very high stringency condition. That is, you can apply high temperature also, but make sure that your high temperature does not actually wash the actual probe from the actual sequence, okay, directly, because otherwise you will not get any signal. So it has to be very carefully done, the washing process. So the membrane is washed to remove unbound radioactivity, and regions of hybridization are detected autoradiographically by placing the membrane in contact with X-ray film. So that completes your, so if you get a band, then you will be able to make out that yes, okay, my genome has the gene of interest. So hybridization under conditions of relatively low stringency permit a high rate of hybridization, but if you go for high stringency, increasing stringency, then it can be done at high temperature or more commonly lower ionic strength. So this is how it looks like. If you see a southern blot, okay, it will look like this, that see, in this case you are getting distinct band, which means that your gene of interest is there. But in certain cases you can see two bands, in certain cases you can see one band. One band means the gene is present in single copy. If you have two bands, that means the gene is present in double copies, two copies. Or if you have one band, then one copy. So in that way you can detect the copy number of the gene in the genome. Next we come to northern blotting. Northern blotting experiment is basically, the technique wise it is actually the same as southern blotting. The only difference is that in northern blotting we use this technique in order to monitor the expression of gene. That is the second step. Once you construct the transgenic plant, you need to find out whether the transcript is being generated, whether the gene is being expressed. For that you go for northern blotting. This was a technique which was used, or which was discovered by Alvin in 1979. So it is used to monitor the expression of gene, and it is used for the comparative expression of genes and estimate the abundance of the transcript. How much transcript is being generated? What is the level of the mRNA? Because transcript basically means the messenger RNA. So how much RNA is being produced? So you are analyzing at the RNA level, and in southern blotting you were analyzing it at the DNA level. So the difference is that now you need to isolate not the DNA but the RNA. So you can isolate total RNA extraction, and then you can either use the total RNA for the northern blotting experiment, or you can use the messenger RNA. If you want to purify the messenger RNA, then you have to pass the total RNA through oligo-DT column, oligo-DT cellulose column, where only the mRNA will be trapped, because the mRNA, it has the poly-A tail. So there is an AT binding, because A base pairs with T. So you have to use an oligo-DT cellulose column. But you can even use the total RNA also for the northern blotting. So basically the process is the same like southern, but the difference is that you need to transfer the RNA. You need to load the RNA in an agarose gel, okay, and then you need to transfer the RNA to the nylon membrane or the nitrocellulose membrane. And then the process is the same. That is, you will prepare a probe, that is, you will prepare a labeled DNA or labeled gene of interest, and you will hybridize that gene of interest with your RNA. So if the transcript is generated, if there is transcript, then ultimately the gene of interest which you have used as a probe will bind to the messenger RNA, so that you will be able to find out that the messenger RNA transcript is generated. Alternately, you can also compare between samples. For instance, I give you 3 samples and I ask you which sample has the highest expression of the gene. So what you can do is you can isolate RNA from the 3 different samples, load them in agarose gel, and then you use the probe for binding and see what is the extent of binding. If the extent of binding is high, then you will be able to make out that the gene is expressed in more, that is, there is a higher expression, and if the level of binding is less, then the expression is low. So in that way you can compare also. Comparative expression can also be done through northern blot. Of course, it needs to be remembered that you need to load the equal amount of RNA from the 3 different samples. That is, you need to estimate the RNA, okay, and that you are loading the equal amount of RNA in all the lengths, in the 3 lengths of the agarose gel. That has to be ensured. Another point which you need to be careful, that in case of RNA gel, okay, although you are running an agarose gel, but you have to make sure that you have to pre-treat the RNA with formaldehyde and dimethyl sulfoxide. and then you need to electrophorese the RNA in formamide agarose gel. So it is not an ordinary agarose gel, it is a formamide agarose gel, or you have to use formaldehyde. Why? Because you have to remember that RNA has a lot of secondary structures. So formaldehyde, it forms unstable sheaf base with single amino groups of guanine residues. These adducts maintain RNA in the denatured state by preventing intra-strand Watson-Crick base pairing. So RNA can be maintained in the denatured state only when formaldehyde is present in the buffer or the gel. So whatever processing you are doing, either with the agarose gel or in terms of the sample preparation, you need to use the formamide or formaldehyde, because it will eliminate the secondary structures of the RNA, and it will allow the RNA to be maintained in a denatured state. Now we come to the third step, that you have generated the transgenic plant, and finally now you want to check whether the protein is being expressed or not. So in this case you need to undergo Western blotting. So this was a technique which was discovered by Taubin, Julian Gordon and Theophil Stalin. Now what do you do in case of Western blotting? So here what you need to do is you need to isolate the protein, not the DNA, not the RNA, but you need to isolate total protein from the same plant, that is transgenic plant. Okay, and now in case of protein we normally don't go for agarose gel, we go for the SDS page, that is sodium dodecyl sulfate polyacrylamide gel electrophoresis. So you run the protein in an SDS page, or you separate the protein bands via the SDS page, okay, and you stain the gel with the Kumasi brilliant blue and you observe the protein bands. Next, the process is almost the same, that you need to transfer the protein, okay, to a membrane, because you are again preparing a blot. You can transfer it again to a nitrocellulose membrane, nylon membrane, or sometimes we also use PVDF membrane for Western blotting. So the three steps are: first you isolate the protein, then you load the protein in SDS polyacrylamide gel, and then you transfer the protein from the polyacrylamide gel to the membrane. Now what you do is you have to have one antibody, because we know an antigen, in immunology we know that an antigen, it binds to an antibody. So what you have to do in this case is you need an antibody which will bind to your protein of interest. Supposing you want to check BT transgenic. So normally the BT protein is there. So you need to have an antibody against that BT protein, against that cry protein. Okay, so you should have that antibody, and then you try to incubate the blot with the antibody. So in this case you are not using any radioactive probe, you are using an antibody against the protein of interest. Now again in this case you have to be careful that you need to eliminate the non-specific binding, and for that you have to incubate the blot with a competitor protein, which in most of the cases we use BSA, that is bovine serum albumin. So you incubate the blot with this protein first, and then you add a substrate buffer, and then you add this particular antibody. Okay, after it is being blocked, you add the primary antibody. So if there is the protein being accumulated, then this primary antibody will bind to the protein of interest. Then what you do is you apply a secondary antibody which will bind to the primary antibody, but this secondary antibody is linked to some reporters, maybe one enzyme linked antibody. Why you are using this enzyme linked? Because after the secondary antibody is added, then finally you need to develop the band, and for that you can apply a particular substrate which will be utilized by that particular enzyme, and you get a colored band. When you add a particular substrate, then this enzyme will be able to degrade that substrate and generate a colored band. So in that way, by visualizing the band or visualization of the band, you will be able to make out whether the protein is being accumulated or expressed. So I just go through the technique: transfer of electrophoresed protein from a polyacrylamide gel to a membrane of nitrocellulose or nylon. Then you use the antibodies Normally in Western blot, we don't go for labeled probes. Normally we go for the antibodies. And then here you are actually using a sandwich approach. That is, single preparation of labeled second molecule can be employed. You can apply a secondary antibody. Sometimes the secondary antibody can be labeled also, but normally we go for enzyme linked secondary antibody. So this is actually the steps. That is, as I already discussed, you run the gel, then transfer it, blot, then block the membrane, then wash, then apply the primary antibody, wash it, then apply secondary antibody, and finally that secondary antibody is linked to some reporters, and then you add the particular substrate, and if the reporter is an enzyme, the enzyme will actually degrade the substrate and therefore there will be band. So in that way you can find out, even you can compare the expression of different proteins or expression of protein between different samples, just as you did in northern blot, you can do it here also. So these are the three very important techniques that you use in the analysis of the transgenic plants, and it is almost a routine work in almost genetically engineered labs, and one has to learn these techniques very properly. Next we go to the DNA fingerprinting, another very important technique. In animal system or in animal world, we know that DNA fingerprinting is very commonly used in case of certain, like, you know, in case of criminal cases or in case of paternity disputes. We normally go for the DNA fingerprinting. We use this technique to solve these problems. But in the plant system also, DNA fingerprinting is very much routinely used. And it was first discovered by Alec Jeffreys in 1984. Certain sequences of highly variable DNA known as mini satellites, which do not contribute to the function of genes, are repeated within genes. Mini satellites are repeated within the tandem repetition of a short motif that spans 0.5 kilobase to several kilobases in length. And these mostly show polymorphism. That means there is a variation in the number of the repeats. So they can cross-hybridize with similar loci throughout the genome, and therefore they can be used as fingerprinting. So fingerprinting is nothing but it's a mode of identification. So it is used to isolate and identify variable elements within DNA sequences, and this is routinely used in forensic science and in paternity dispute cases, especially in animal biotechnology. So what are the steps? You isolate the DNA. So the first steps, the initial steps are similar like southern blotting. You isolate the DNA, digest the DNA with enzyme, restriction enzyme, then separate it through electrophoresis, blot the separated fragments onto membrane like nylon membrane, and now what you do is you hybridize the fragments using specific probes. What are those probes? These are called VNTR, that is variable number of tandem repeat. So you use these VNTR probes for binding, and then again you have to develop the blot through autoradiography, and then you go for the analysis of the hybrid fragments. So you see it has similarity with southern blotting. Only difference is that the choice of the probe, that is different in this case. Now in plant biotechnology, we have one very important topic called marker assisted selections, or we use certain DNA markers. Now the two such very common DNA markers which you have in the plant biotechnology are RFLP and RAPD, restriction fragment length polymorphism, that is RFLP, which was discovered by Botany and co-workers in 1980, and RAPD. So I first take RFLP. Now what are the applications of RFLP? In fact, there are several applications, and I have listed only very few of them, like construction of linkage map of any genotypes and genes linked to certain specific agronomic characters are successfully mapped by RFLP markers. In certain disease management, like you want to detect certain pathogens, like RFLP based on VNTR from genomic library of Pythium has been used to identify the plant pathogen Pythium. Analysis of somoclonal variation. Now I hope that you know the meaning of somoclonal variation. What is somoclonal variation? That is, in course of tissue culture, when you are culturing any plant, like when you go for genetically engineered plant also, you have to go for transformation, plant transformation, which I discussed in my earlier sessions, that plant transformation through agrobacterium tumefaciens mediated transformation. and or you can go for the gene gun or particle bombardment. But the thing is that while you are undergoing this culture process, there can be certain variations, and these appear spontaneously. There can be certain variations which you might come across when you are culturing, when you are going for tissue culture or cell culture. So these variations are called somoclonal variations. Some of the somoclonal variations can be very useful. In fact, many desirable traits that we have got, okay, economic traits that we have got, they have come directly from somoclonal variation. But sometimes again they might be detrimental. They might lead to the plant death. Okay, so it depends. So these somoclonal variations, you can use the RFLP molecular markers for detection of this genetic variation and prediction of genetic distance between possible parents. How close the parents are, that can also be predicted through RFLP. So what are the steps in RFLP mapping? First, you need to select the parent. First step, they should be selected that they exhibit polymorphism, maximum polymorphism, mapping population and size. Different mapping populations can be used. Usually it is the F2 population derived from F1 progeny or backcross population which is selected. So the size of the population is very important. Then you need to isolate the DNA. Now the process is almost the same, because you know RFLP is also, you can say that it's a kind of fingerprint also, you can say. So restriction digestion, high quality DNA must be isolated from the parents and the F1 and F2 population before it is used for restriction digestion. Then you need to separate the digested DNA, that is again you can undergo the agarose gel electrophoresis, and then here you apply the southern hybridization, that is separated DNA is transferred to nitrocellulose membrane. Single copy label probes such as cDNA or EST. EST stands for the expressed sequence tags. So you are using these labeled probes for hybridization, that is for binding. The developed autoradiogram reveals the banding position. Scoring for the presence and absence of DNA band is done for the parents, the F1 progeny and the F2 progeny. So you can find out what is the source. Okay, you can compare the parent, the F1 progeny, the F2 progeny, you can compare the profile and then you can find out how close they are. Even you can check the evolutionary distance or evolutionary affinity using this RFLP technique or RFLP mapping. Construction of RFLP map using computer program. Based on the data collected, the likelihood of linkage and absence of linkage between markers can be calculated to obtain the logarithm of the odds, that is LOD score, in order to construct the genetic map. Actually, LOD score is nothing but it gives you an idea of the evolutionary affinity, how close they are. Since the data is large, a computer program has been developed to do it, and in this case, the most important or very popular program for genetic map construction is termed as MapMaker. So using all these tools or using several statistical tools and computer softwares, you can actually construct RFLP map, that is restriction fragment length polymorphism map. So this is actually the profile that that they have shown in RFLP profile. You can see that is lane number 2 and lane number 5. They are typical pattern, but there are variations you can see in lane 1 and lane 3 because they are having one extra restriction site. And lane 4, as you can see, it's more variation. That is, they show a variant of line 3 with one extra restriction site. So you see, you can compare between the different lines, okay, based on the RFLP profile. Next we go to the another DNA marker, that is RAPD, that is randomly amplified polymorphic DNA, which was identified by Williams and co-workers in 1990. Now here, what are the applications of RAPD? Distinguish between variety. That is, like for instance in sunflower, almost 15 commercial varieties have been identified using this RAPD. Construction of genetic maps. RAPD markers have been used to construct 15 linkage groups in coffee. Then selection of segregating populations during plant breeding. Selection of desirable trait. Molecular marker linked to the trait of interest can be screened. And finally, they are also used for the identification of desirable trait in long-lived species, which takes long time for maturity and show phenotypic character. In case of Puccinia graminis, stem rust, that is RP94 gene, which is responsible for resistance to stem rust in barley, that has been identified through RAPD. Okay, and several RAPD markers have been linked to this particular gene. In tissue culture, somatic hybrids involving protoplast fusion. So this is a very popular technique. We call it somatic hybridization, where you can fuse protoplasts from two different species or even from two different genera. It is possible, and you can create a somatic hybrid. So mostly we use this technique, protoplast fusion technique, where the traditional crossing or traditional hybridization process fails. So there we try to fuse the protoplasts and see if you can generate a hybrid. So obviously the hybrid profile, the RAPD profile of the hybrid will not be the same like the source protoplasts or the donor protoplasts. So you can compare. But again, there will be some similarity. There will be similarity with one donor and there will be also similarity with the other donor. So you can make out whether you have actually been able to create a somatic hybrid properly based on the RAPD profile. So it is also used as a, it's also used for certain diagnostic marker, that is identification. You can use this particular technique. RAPD analysis provides an important tool for the characterization of biodiversity. Now, how does this process work? The difference is that here, why it is called randomly amplified? Because you use random primers. You don't use any gene specific primer, because immediately I will go to the technique of PCR, that is polymerase chain reaction, where you will see that we use gene specific primers mostly. But here you are using random primers. Okay, so it detects nucleotide sequence in a DNA amplification based assay using only a single primer of arbitrary nucleotide. A single species of primer binds to the genomic DNA at two different sites on opposite strands of the DNA template. If these priming sites are within an amplifiable distance, a discrete DNA product is produced. Polymorphism between individuals result from sequence differences in one or both of the primer binding sites and are visible as the presence or absence of a particular RAPD band. So you can compare between different samples and you can check which band is present where, which band is missing, which band is present. You can compare between different samples. So therefore we call it a polymorphism, because you may not find the same profile in all the samples, so that there are polymorphisms. So such polymorphisms are actually being used or made use of in the RAPD technique. So you have the extraction. Again, you need to isolate the DNA and you need to have the random primers, and you have to just go for PCR using the random primers. And then you can run the agarose gel, and you can see from the figure that you have a monomorphic band. Monomorphic means which is the same in both the samples A and B. But polymorphic band which may not be present in one sample. Like in A, you have one band, but in B, you don't have that band. So that becomes the polymorphic band. And therefore, this is a technique which is used to detect polymorphism. So this is one of the standard profile. Okay, people have run agarose gel. Okay, and this is how an RAPD profile look like when you compare it among different samples. Like here you can see as many as 36 samples, and they are showing the RAPD, comparative RAPD profile. Next we go to two very important techniques. These are very much allied techniques. One is PCR and another is RT-PCR, that is polymerase chain reaction and reverse transcriptase polymerase chain reaction. Now polymerase chain reaction, Kary Mullis in 1983, he got the Nobel Prize for the discovery of the polymerase chain reaction. Now in PCR reaction, what are the components that you need to use? That is DNA template, DNTPs, forward and reverse primers, 17 to 30 nucleotide, and the unique part is that you are using a heat stable Taq polymerase, which is isolated from one thermophilic bacterium, okay, Thermus aquaticus, which actually lives in hot springs or resides in hot springs, and it survives temperature of about 90 degrees centigrade. Now the reason is that why you are not using normal DNA polymerase for amplification is that because you need to undergo a denaturation step in the PCR reaction, and that denaturation step is almost 94 degree or 95 degree centigrade. And at such high temperature, the enzymes will not be able to maintain their activity. So you need to have a heat stable enzyme, and therefore you use this stack polymerase. So here what you do is there are 3 steps. First step is the template denaturation step. Second step is the annealing step. Annealing step means the primer will actually bind to the template. So what is the purpose of doing PCR? Again, if you want to amplify a particular gene, like you need to clone a gene. So what you have to do, you have to amplify the gene from the genome. So you need to have the gene specific primers, and you can, through PCR, you can amplify the gene, or you can get the gene or derive the gene from the genome, and then you can clone the gene. So that is one of the utilities of PCR. Another utility is that you can even confirm through PCR that whether the gene of interest is present in the genome or not. That is, the purpose is the same like southern blotting. Okay, so even the thing that you got through southern blotting, conclusion that you got through southern blotting, okay, regarding the integration of a gene can also be derived from the PCR or polymerase chain reaction. So denaturation, then the primer annealing. Primer has to anneal to the template, and then the DNA synthesis. And there are several cycles. Polymerase chain reaction. Why chain reaction? Because you need to carry it under different cycles, as many as 30 to 40 cycles. So people have shown that 30 cycles, if you undergo PCR for 30 cycles, then you get the maximum amount of template. So primers hybridize to opposite strands of DNA after it has been denatured and are oriented so that DNA synthesis proceeds through the region. Extension reaction creates 2 double-stranded target regions, and the third cycle produces 2 double-stranded molecules that comprise precisely the target region in double-stranded form. So there are repeated cycles, as many as 30 cycles, and each has the 3 reactions, that is, as I told you, the denaturation, the annealing, and the DNA synthesis. And after 22 cycles, there is an amplification of about 106-fold. So you can get an amplified gene, and you can use it directly for the cloning purposes. So that is actually the real advantage of undergoing PCR reaction. You are amplifying a particular gene so that you can get it in a considerable copy number, and you can clone the gene. Now, reverse transcriptase polymerase chain reaction, or RT-PCR, the technique is basically the same like PCR, but here what you do is you have to isolate the RNA. So the purpose of doing RT-PCR is the same like northern blotting. That is, you need to monitor the gene expression. So what you do in this case, you isolate the RNA, and then using the reverse transcriptase enzyme, you convert the RNA to the cDNA or the complementary cDNA. So the primer annealed to the RNA of interest is usually a synthetic oligo-DT or a synthetic oligonucleotide complementary to a specific transcript, and this DNA-RNA hybrid will serve as a template for reverse transcription. So you use the reverse transcriptase enzyme, and you get, using the cDNA, you get the desirable target gene of interest. That is, whether the gene is expressed or not can be confirmed through RT-PCR. I have 2 more techniques to discuss. One is Sanger sequencing method, and another is the ELISA. Now, sequencing. Nowadays, we are very much in, whether in plant science or in animal science, we are very much familiar with this technique, that is sequencing. Like once you have cloned a particular gene, okay, you have cloned a particular gene in any bacteria or even in plant, you need to sequence it. Or like you have cloned a gene which is unknown gene, you need to sequence it, because otherwise whether you have cloned the right thing or not, that has to be understood, and for that you need to know the gene sequence. So a lot of techniques, high throughput techniques, are nowadays available, but the first and the foremost technique which was, you can say, which was discovered, and most of the modern techniques are actually based on that technique, is the Sanger sequencing method or Sanger-Coulson method. Now this method is also known as dideoxy-mediated or chain termination method. It involves the use of chain terminating dideoxynucleoside triphosphate. Mind that this is not DNTP, this is DDNTP. DNTPs we use in a polymerase chain reaction or even during the replication DNTPs are used. But here we are using something different, that is DDNTPs. Now what is the difference between DDNTP and DNTPs? The DDNTP differ from DNTPs in that they lack a hydroxyl residue at the 3' position of the deoxyribose. So they can be incorporated by DNA polymerase into a growing chain through their 5' triphosphate groups. So this is the principle. So basically it's also called as the chain termination method. Why is it called chain termination method? You will be able to understand right now. The absence of a 3' hydroxyl group prevents the formation of a phosphodiester bond with the succeeding DNTP. Further extension of the growing chain is then stopped or terminated. So whenever it encounters DNTP, then there is a chain termination. Thus, when a small amount of one DDNTP is included along with 4 normal DNTPs in a reaction mixture, there is a competition between extension of the chain and infrequent but specific termination. So when you are setting up a reaction, you have to mind that you are using the DNA, you are using a primer, okay? It can be universal primer, T3 and T7 primers or M13 forward, M13 reverse primers. These are called universal primers. And of course, those primer sites has to be there in the vector. And you have cloned the gene of interest within a specific vector, having those universal priming sites. And then you apply the DNA polymerase and the 4 DNTPs, of which 1 DNTP is radioactive. And then after this mixture is prepared, you separate them into 4 different tubes, and in each tube you add one specific DDNTP. So you have this mixture and you separate them, and then you add, like you can see in the diagram, you add the different DDNTPs. Okay, so if DDNTP is present, as I told you, synthesis will stop when this molecule is incorporated into the growing chain. So you have 4 separate reactions. In each reaction you have a separate DDNTP, and in addition you have all the 4 DNTPs. So the ratio of the DNTP to DDNTP is carefully adjusted so that the dideoxy moiety is incorporated only occasionally in place of its deoxy homologue. And mind that when you are done with the reaction, then again you have to run a polyacrylamide gel. Why in this case you are not using agarose gel but a polyacrylamide gel? Because you need to identify each and individual fragment. You need to identify each and every sequence. And normally a sequencing gel, when you develop an autoradiograph, because as I told you, one of the DNTPs is radioactive, like you use DATP. So automatically you are using a radio-labeled DNTP, and when you develop the autoradiography, you will be able to see the bands, okay? And these bands has to be read from below, not from the top of the autoradiograph. You have to read the sequence from below, like as you can see in this diagram, as they have shown, that sequence reads from this, like this is G. G corresponds to, as I told you, DD-GTP. C means this reaction is DD-CTP. This one DD-TTP, and so on. So G, like this sequence will be like first G, then A, then another A, then it will be T, and so on. So in that way, Sanger sequencing is being done. Lastly, we come to the technique called ELISA, or the enzyme-linked immunosorbent assay. Now this technique is very much commonly used in medical science, okay, because this is actually an immunological technique, but in plant science also we use ELISA, okay. So an enzyme conjugated with an antibody reacts with a colorless substrate to generate a colored reaction product. Such a substrate is called a chromogenic substrate. A number of enzymes have been employed for ELISA, including alkaline phosphatase, horseradish peroxidase, and beta-galactosidase. So this technique was again developed by E. Engvall and P. Perlman in 1972. Now this ELISA, it can be of 3 different types. First one is called as the indirect ELISA. Now what happens here? the serum or some other sample containing primary antibody is added to an antigen coated microtiter well. So here it's microtiter well and antigen is coated. Then after any free antibody is washed away, the presence of antibody bound to the antigen is detected by adding an enzyme conjugated secondary anti-isotype antibody which binds to the primary antibody. Any free antibody is then washed off and the amount of colored reaction product is measured by spectrophotometric plate. So in this case, antigen is coated and you add the antibody. Sandwich ELISA, it's just the reverse. Here the antibody is coated. That is microtiter well, you coat it with antibody. So, okay, and then it is immobilized and then you apply the antigen and allowed to react with the immobilized antibody. Then you go for the washing and you use the second enzyme linked antibody for a different epitope on the antigen. Lastly, in case of competitive ELISA, this is another variation. Here what you do is you first mix the antibody in a solution with a sample containing antigen. You have the antigen antibody mixture and then you add it to a antigen coated microtiter well. So if there is more antigen, there will be more binding of the antibody and therefore the free antibody will be less available to bind to the antigen coated well. And then you apply or add the enzyme conjugated secondary antibody which is specific for the isotype of the primary antibody. So this is a competition. Basically there is a competition. Okay, and that is why it is called as the competitive ELISA. So, so today we have actually discussed these techniques, some of the very important techniques which are routinely used in plant biotechnology, and of course techniques like PCR, RT-PCR, sequencing, southern blotting, northern blotting, western blotting. These have to be very, very carefully understood because as I told you, these are routinely used in molecular biology, in plant molecular biology, especially when you are going for genetic engineering. Okay, and in the breeding, plant breeding areas, mostly we use the markers like the techniques that I discussed, that is DNA fingerprinting, then RAPD, RFLP. These are more commonly used in the plant breeding areas, but in transgenics, mostly we have the PCR, RT-PCR, southern blotting, northern blotting, western blotting, fingerprinting, sequencing. So these are the techniques. So please go through these techniques very carefully because at the graduate level, because even if you later on go for the postgraduate studies, you will need these techniques. Maybe you need to learn this. You can also have a hands-on training of some of these techniques like PCR and RT-PCR. You can also go for the hands-on training. And these will be very much required to learn, okay, the protocols and the basic principles of these techniques. Okay, so that is all for today. Thanks for your hearing, and I wish you all a very, very happy learning. Please go through the units, and in case of any queries or any doubts, you are always free to contact us. You can write to us and or you can call us. You can give your valuable feedbacks. So that is all for today. Thank you.